Oxidative phosphorylation. This is the topic for today, and this step is the last step of respiration. That means after the glycolysis, Krebs cycle, and the electron transport chain, oxidative phosphorylation is the last step where ATP are produced using the NADH2 and FADH2. So, what is happening actually is in glycolysis and Krebs cycle, the NADH2 or FADH2 which are produced, they dissociate to produce the electrons, and these electrons move through the electron transport chain in the inner membrane of the mitochondria. While they move through various electron acceptors at different levels, they will release energy as we have studied in the previous videos. And this energy which was released was used in pumping protons from the matrix of the mitochondria to the intermembrane space. Now let us try and understand and go back to the situation. This is the outer membrane. So here I write outer membrane. Then this is the inner membrane which extends into foldings, and these foldings are known as cristae, which extend towards the matrix side. So this will be the inner membrane. This is the intermembrane space where protons will be released. These are the cristae of mitochondria, and this area, cristae, opens into matrix. So, recalling whatever happened, the glycolysis and the Krebs cycle, the NADH2 and FADH2 which are produced, they release electrons because these activities are taking place over here. So, the electron transport chain actually will be taking place over in the inner membrane of the mitochondria, and the electrons are energy is released, and as a result this energy is utilized to pump hydrogen from the matrix to the inner or the intermembrane space. Now we know that one electron or the release of the pair of electrons are causing the movement of two protons. One NADH2 will be producing three protons. So the protons are therefore in higher concentration in the intermembrane space over here or the outer compartment. Now these protons have to move from high concentration to the low concentration, because continuously with the movement of the electrons through the electron acceptors, the protons are being pumped from the matrix to the intermembrane space. As a result, the concentration of the protons is reduced in the matrix, and the tendency of these protons will be to go back to the matrix, and during this movement, that is going back from higher concentration to lower concentration, they will be releasing energy during this transfer. Now these protons can move through this membrane, or they can go from high concentration to the other concentration, that is towards the matrix, through particles which are, this is the F0 which we have studied, and this is the stalk, and this is the F1 particle. So we have F0 over here, this dark is the F0 particle, F1 particle, and this is the particle through which the F0 F1 particle through which the electrons will be passing, and subsequently will entering into the matrix. So the F0, F1 particle, that is the particles which are present towards the matrix in the crystal, these particles act as the gateway or the channel for the proton to enter into the matrix. Now, when the proton is being transferred, the proton has certain amount of energy. And when it passes through, it will pass through, the F0 is embedded in the inner membrane, as you can see over here. And when this hydrogen ion or the proton having energy passes through the F0, and when it passes through the F1, it will release certain amount of energy. Now this F1 particle also act as ATPase, that is the enzyme which is used for the synthesis of ATP. So here what will happen, as the protons will be passing through this F1 particle, the ATP will be produced from ADP and the inorganic phosphate. They will combine in this F1 particle, utilizing the energy which is released by the protons as it passes from the intermembrane space to the matrix, and ATP will be produced. So we find that ATP synthesis taking place. Now, as a result, what is happening over there? ADP and inorganic phosphate are combining in presence of ATPase to produce ATP. The proton gains the entry back into the matrix. Now, if we recall, or let us summarize this, what is actually happening is the glycolysis process, the Krebs cycle, and they will release the NADH2, which further releases the electrons when it dissociates and releases energy during this dissociation. This energy is utilized to pump the protons, a pair of protons, every time from the matrix to the intermembrane space, that is the space between the outer membrane and the inner membrane. As a result, because ETC, that is the electron transport chain, is taking place within the inner membrane. Now, due to this, the concentration of the protons will obviously be higher in the intermembrane space, but the matrix will have a lower concentration. Also, the tendency of the protons having certain amount of energy will be to go back to the matrix, and this can be done only through the F0 F1 particle, which is the channel through which the protons carrying energy will pass through this and will enter into the matrix to balance. Now, as the protons carrying certain amount of energy shown by the wavy line passes through the F0 particle, and when it enters into the F1 particle, it will release certain amount of energy, and this energy which is being released by the protons is utilized to combine the ADP, adenosine diphosphate, with inorganic phosphate to produce the ATP. Now, so if we see over here from one NADH2, there are three pairs of protons being produced during the movement, or during the dissociation, and this pair of protons are produced and are pumped to matrix over there, and matrix to the intermembrane space, and one pair of proton will produce one ATP over here. We have seen one pair of proton, as it passes through this, it will produce one ATP. That means we have since one NADH2 is producing three pairs of protons, that means one NADH2 is equivalent to 3 ATP, because it is worth 3 ATP. It produces 3 ATP. And cristae is the site where actually the ATP is produced, and we are using NADH2 or FADH2 produced during the previous processes. We are using that this currency, or we are caching this to produce ATP. Now here you have to understand the difference between the NADH2 and FADH2, which we are continuously talking about. The difference between NADH2 and FADH2 is that when NADH2 dissociates, the electrons will enter into the electron transport chain and it will produce energy at three different levels. We have studied in the previous video. So energy released by the NADH2 dissociation is at three levels, and at each level a pair of protons is produced. Now a pair of protons will be produced, and one proton is equivalent to 1 ATP, because when the transfer will take place from the intermembrane to the matrix. So 1 NADH2 is equivalent to 3 ATP. Now let us compare about the how the FADH2 is different. When we talk of FADH2, 1 FADH2 during its dissociation produces only or releases energy at two levels. So it produces only two pair of electrons. Two pair of electrons will be produced, and as a result we know, two pair of protons. Two pair of electrons which will pass through the electron transport chain, and as a result two pair of protons will be produced. So 2 into 2 we have two pair of protons, and they also will release energy in the form of ATP, and that means 1 FADH2 is worth 2 ATP. So we find NADH2 produces more ATP or more energy. Now we will be calculating the energy produced during the entire respiration process in the form of ATP, that is the balance sheet of the aerobic respiration.